All right, so today we are going to talk about meiosis. We're going to cover a little bit about the difference between meiosis and mitosis, and we're going to talk about why meiosis is so important for the continuation of a species. So we're going to start off with some vocab words. So our first thing that we need to think about is how is it possible for a child to get information from both parents? So when you think about it, your DNA or genetics comes from mom and it comes from dad. And if we know in humans, we have 46 chromosomes. But if mom passes her 46 chromosomes and dad passes his 46 chromosomes on, we are not going to end up with 46. So, what do? How do we figure that out? Well, that's where meiosis comes in. Remember, I know it was a long time ago that we thought of this, but in mitosis, we make two identical daughter cells. In meiosis, we are not going to end up with two identical daughter cells. We're going to end up with something a little bit different. So, we have our first vocab word that is diploid. Diploid is a cell that contains both sets of homologous chromosomes from parents. So our homologous chromosomes means a chromosome from mom and dad. They contain the same traits from each parent. So if you have one trait for hair color on one chromosome, hair color will be on the other chromosome. That does not mean they have to be identical. So you can get one trait for brown hair and one trait for blonde hair. They can be different, but they will be the same trait. So there are matching chromosomes from mom and dad. In humans, you have 23 from the sperm cell, which is in males, and 23 come from the female and egg cells. We're going to symbolize this as 2N, which is found in body cells. And N is going to be our number of homologous pairs. So human 2N is 46. Drosophila, which is a fruit fly, 2N is 8. So this right here is a quick picture of meiosis showing haploid numbers. So our haploid number, haploid is half. So in our sperm and our egg cell, we have 23. Fertilization, when we make that diploid, we have 46. We have our multicellular diploid adults, 2N equals 46. This is mitosis and development. So we are growing from that original cell, from a cell to eventually turning into a human being. From there, we can undergo meiosis. And then we can go back into from mitosis to meiosis. So haploids are cells that contain one set of chromosomes or germ cells. No big deal if we remember what germ cells are. And the symbol is just an N. Oop, it came up kind of weird. But in humans, N is 23, Drosophila, N is 4. So diploid is 2N, haploid is just an N. The definition that we're going to use for this purpose is that meiosis is reduction division. So the number of our chromosomes is cut in half. So the homologous chromosomes, so our pairs of chromosomes, are split in half and they're going to make sex cells. So if you start with 46, you end up with 23. That's all you need to know. So we have right here a cell that is undergoing meiosis. You have the same exact names of phases. So you have metaphase, they're lining up in the middle. Anaphase, they're moving away from each other. Telophase, splitting apart. Then you have another set of anaphase, another telophase, and again, you notice that it is 4 cells. So the difference in meiosis is we go through 2 stages. So interphase is the same as exact thing in mitosis. Chromosomes are duplicated. I'm going to attach these 2 web animations. I suggest that you watch these. I'm not going to talk through them. I'm going to let you guys watch these on your own. I'm sure you are sick of hearing my voice on these videos. I know I'm sick of talking through things, so I'm going to let you just listen to another person talk to you. The first one of cell division includes prophase I, metaphase I, anaphase I, telophase I. You can tell they were very, very, very creative when they came up with these names, made it so much easier. But we have this thing, it's called crossing over. So if you notice right here, I'm going to try to grab a pointer to make it a little bit easier. So they named these segments on our chromosomes. So remember, these are duplicated because they're in the shape of the X. So they named it like capital A through capital E, then lowercase a to lowercase e. So crossing over is when you have a pair of homologous chromosomes and they just flip-flop bits of their genes. This increases genetic diversity. So if you have a sibling, you look a little bit like your sibling or a lot of it, but you're not identical to your sibling. I have a younger sister and I feel like I look nothing like her. I have brown hair, she has blonde hair. I'm tall, she's short. I'm tan, she's pale. I'm better, she's not so much better. But it's the idea of crossing over occurs to give those differences. And down here we have what's happening in interphase. We call this a tetrad. A tetrad is just a pair of homologous chromosomes. Usually they line up nicely by size. We start with tall and then we go to the short ones. Metaphase I, same exact thing as metaphase in mitosis, except instead of individual chromosomes lining up, we have the homologous pairs finding their buddies and lining up. So in each new daughter cell in the first split, we can pull them apart. So spindle fibers still attached to the centromeres. Remember, centromeres are in the center of our chromosomes. And then the homologous pairs line up in the center of the cell. Another little animation. In anaphase I, our spindle fibers pull the homologous chromosomes apart from each other. Remember, anaphase away. What I really like in these animations is you can see that there was crossing over that happened because of the colors that you see. So there was some crossing over evidence that's been happening. Telophase I, same thing that's happening. Cells start to pinch off. Our nucleus is going to reform. Chromosomes uncoil. Another video I'm going to post underneath the notes to watch. Prophase II, we have our centrioles, our spindle fibers forming. Nucleus is disappearing. Now our cells are haploid, so they are just N cells. Chromosomes become visible again. So we don't have our pairs of chromosomes anymore. We just have the single X's. These are still, remember, duplicated chromosomes, still duplicated. Metaphase II, our duplicated chromosomes line up at the center. They're moved again by spindle fibers. Anaphase II, they get pulled apart, same as in mitosis. They move to opposite ends of the cell. Telophase II, our nuclear membrane forms. Cell starts to pinch off. Our chromosomes uncoil. And now we have 4 haploids, so we have 4 N cells are created. So males versus females in meiosis. In the background, you can see you have this egg versus the sperm. The first thing you can see is that the egg in comparison to the sperm is huge. When males undergo meiosis, they will make 4 identical sperm cells. When eggs undergo meiosis, they will produce 1 usable egg and 3 unusable polar bodies. What happens is all the cytoplasm goes to 1. So basically, you have the mother splits. This just doesn't do anything. It's non-functional. Basically, all the cytoplasm goes to 1. Splits again, nothing. This egg has everything, all the nutrients that's needed. So 1 egg is going to be huge, have all the nutrients that are going to be needed, have everything that's there. Also, another thing to know is that the egg will also have all of the organelles needed. So something that's really interesting is you get the mitochondria. Mitochondria have their own DNA within them, because in evolutionary history, go off on a little tangent, mitochondria used to be their own living organisms, and then they were absorbed by eukaryotes. So mitochondria have their own DNA, and you can actually trace lineage of mothers based on mitochondrial DNA because the mother has DNA in there, and you have that DNA from your mom in your mitochondria. The sperm only has DNA, only has a nuclear DNA because they are small, they make four. It also takes a lot of work to get into the egg, so it makes sense. We're going to look quickly over the difference between mitosis and meiosis. So the type of cells for mitosis is body cells. It's going to happen in your skin cells, bone cells, muscle cells, stomach cells, everywhere. Meiosis only happens in germ cells, only sperm and egg. Number of cells produced, that's going to be 2. Meiosis, that's going to be 4. Type of cells that are produced, diploid, remember that is 2N. We have the copy from mom and copy from dad. In meiosis, that's our haploid cells. That is half the number of chromosomes. In cells produced compared to the original, mitosis, you have the exact same thing, genetically identical. In meiosis, they are genetically different. Remember, we have crossing over, and then we have the way that the tetrad, so the pairs of homologous chromosomes, get pulled apart. That's going to cause a lot of variation, so you're going to be different than your siblings. Number of divisions, there's only one division in mitosis, and in meiosis, there's going to be 2 different divisions. Crossing over does not occur in mitosis, but it does in meiosis. All right, so that's all I have for mitosis and meiosis. If you have any questions about this, please let me know. And what we can do is we can go back over this. I'm going to have you go through this video with the notes, and then I'm going to have you basically do one quick practice and then basically like an open note test with this. So my goal is for you guys to have all of this meiosis, mitosis review done by the end of next week. If you have questions and you want to get on a Zoom meeting, please let me know. I posted my office hours on the Harbor Classroom, and I also have office hours that are available via text or email. You can set them up after school. All right, if you guys have questions, please let me know. I'm always here to help.